So as we move on to the next topic, we welcome Dr. A.J. Joseph, who is the professor and head of Department of Gastroenterology, CMC Vellore. Sir is an exemplary clinician and academician, and he has numerous national and international publications to his name, with special areas of interest in the pancreas and the GIT. So we welcome you on stage to talk about the microbiome and its correlation with chronic kidney disease. Thank you, sir.
Good morning, everybody. Thanks for this opportunity to speak here. I see the person who I first met when I came to CMC, Dr. Chaku Korla Jacob, was my GO for my interview. So let me, I've divided this talk into some basics on the gut microbiome and how it's relevant for us, and then its relationship with kidney disease, and then whether we can do anything to the microbiome to affect the outcome of kidney disease. I'll spend a little bit of time on this, the way we study the microbiome. The number of papers in microbiome, gut microbiome, has really exponentially increased with newer ways of non-culture methods of assessing the bacterial diversity and the numbers in the gut. And when we use traditional methods, only a minority of the bacteria are culturable. If you look at the genes and then you do PCR or the shotgun metagenomics, 16S rRNA studies, you can look at not just bacteria but other non-culturable organisms like archaea, fungi, viruses, other small parasites, bacteriophages. The term microbiome is all the genes that you assess is called microbiome. Microbiota is the term that encompasses all the living organisms in the gut. 90% of them belong to firmicutes or bacteroids, and then there are about 10% of them are, when you look at bacteria itself. There are two concepts here that I want to just spend a little time. When we talk about dysbiosis, we are talking about the change in the diversity of the bacteria in the gut. So you take antibiotics will kill off some bacteria and leave some bacteria. So sometimes we have a patient with diarrhea, you send a stool to microbiology lab, they'll say normal coliforms are not grown in the stool. So that is a dysbiosis. There are two terms used here. One is called alpha diversity, one is called beta diversity. Alpha diversity is the amount of diversity in one sample of stool or one habitat. So let's say you take a group of 50 people from India, and if you see that there are people from 5 states, that is a reasonably diverse group. If you see people from 2 states, it's not so diverse. But out of these 50 people, if 49 are from one state, or let's say 46 are from one state, and the other 4 are from one state each, that's also not good diversity, even though the number of states is the same. If you look at, if these 5 states are all South Indian states, then the diversity is not great, even if you have 10 people from each state. But if you have 10 people from north, 10 people from south, 10 from west, then the diversity is more. So there are, if you think of these 3 concepts, diversity is not just the number of different bacteria, it also means how the bacteria is spread. So there's something called evenness of the bacterial spread. And the third concept is the phylogenetic diversity. So if you look at people from North India, South India, East, they are ethnically diverse and diverse populations. So bacteria also, if you draw it on a phylogenetic tree, you can have phyla which are closely related or phyla that are not so closely related. So that is the third concept. So all these concepts are involved when you say alpha diversity. And there are indices which capture all these three components. So you have the diversity of the sample, the number of bacteria in the sample, whether they are spread evenly, and how phylogenetically closely related they are. So when you have a sample with good alpha diversity, all those components come in. Beta diversity is a difference in diversity between groups. So if I take a group of patients with inflammatory bowel disease, and then I have a group of patients with normal controls, as two groups, if I compare the diversity, that's called beta diversity. But in our studies, IBD is a big thing for us in microbiome studies, but you can do the same thing with soil, you can do the same thing with skin, throat. People have done microbiome studies from deep sea, thermal vents, volcanoes. So the samples are varied. The gut microbiome grows and changes as we grow. So there is a microbiome, amniotic fluid has got a microbiome. The newborn acquires, depending on the mode of delivery, acquires a microbiome from the mother's vagina or the skin, depending on whether it's a vaginal delivery or a cesarean section. Whether the child is formula fed or breastfed, whether the child has received antibiotics, all those affect what your microbiome eventually consists of. As a child, before solid food is introduced, the flora is called a bifidus flora. It's a bifidobacterium dominated flora. And as the child is weaned and goes on to solid foods, it becomes a normal adult flora. So you acquire a normal adult flora by around the age of 3. The normal adult flora is required to handle normal food. Normal food involves a lot of complex carbohydrates. It involves xenobiotics, things which are produced by other organisms. So as the child grows, the bacterial flora also changes, and by about 3, it kind of stabilizes. And generally, it's quite stable and throughout life. If you think of the 3 major functions of bacteria in your gut, I think it can be divided into 3. One is protection from pathogens. You have antibiotics associated diarrhea, Clostridium difficile when somebody is on antibiotics. So your normal microbiota will protect you from infections. It's important in immune regulation because normal bacteria are involved in maintaining the homeostasis and making sure that gut leakiness is prevented, and also metabolic functions. So there is an interesting study where germ-free mice were taken and given stool from 2 groups of patients, 2 groups of people, one a lean person and one an obese person. So the same mice, identical mice, given a lean person's microbiome and a fat person's microbiome, and then they're given the same food. And the mouse which has got a lean person's microbiome grows up to be a lean mouse, and the mouse that has got a fat person's, obese person's microbiome has got puts on more weight. You also see that, I think, sometimes, dogs and owners also look alike. And I feel maybe it has something to do with microbiome. Maybe they share the same microbiome and develop features of, So there is a lot. Your physical characteristics can depend a lot on what your microbiome is. Now, one more basic information to start with is how the bacteria are classified. You know that starting from school time, eukaryotes, prokaryotes, then bacteria, viruses, fungi. So in microbiome studies, sometimes you are able to tell even up to the species level what you are dealing with. Sometimes you are not able to tell up to the species level, but only up to the genus level. There is also strain level, which is even more below the species level. I just put an example which is familiar to all of us because that causes HUS. That's a E. coli O157H1 serotype. So below strain level, below species level, you can have further classification depending on the serovars, the type of. So it's a particular serotype that reacts with the flagellar antigen and the capsular antigen. O antigen is the capsular antigen, H is the flagellar antigen. So that is a strain for HUS. Now, there are many ways of looking at the microbiome. The most common thing that you will see in studies is using the 16S rRNA or the studying the whole basket of genes that are available in the sample. 16S, we'll talk about 16S rRNA, that's what we've used in studies, and that's a cheaper way to do things. So 16S rRNA is kind of unique in the sense that it is present in all bacteria and another group of organisms called archaea. It is a ribosomal protein. The thing about this protein is it is present in all bacteria. So it's got a very highly conserved, gene. But the advantage is it's got a very variable segment also. So it's got two segments. One is a very highly conserved segment and one is a very variable segment. So if you amplify the gene using the highly conserved segment, you can amplify the 16S rRNA. And then if you study the variable region, you can classify it phylogenetically. So you can get groups of bacteria which are grouped really not as species level. It doesn't give you species level resolution, but it gives you genus level resolution. And the output is called taxonomic units. That's the term that you'll come across, OTUs, in 16S rRNA studies. In metagenome sequencing, what is done is the whole sample is, I think with technology getting better, the whole sample, you can look at all the genes in the sample. But the problem with that is it includes sample, like if you're looking at a biopsy sample, it will have genes from the host DNA also. Clearly the output will be the amount of data that you generate is much more. It will be like 300 TB of data when you do a metagenome study. And the amount of computational resources that you require, and therefore the cost is also higher. But metagenome, if you look at metagenome studies, you can identify all bacteria, fungi, viruses, everything, plus you can look at metabolic pathways also. Like suppose I want to see what is the capacity of my gut microbiome to handle, say, urea. So I can look at the genes which are enzymes which are coded by enzymes involved in the pathway and the genes which are coded by the pathway, and I can look at the amount of those genes in that sample. So I can get an idea of not only what bacteria is available, but also I can have an idea of what functions is that like sample, what metabolic functions does it have. So that is the advantage of it's called shotgun metagenomics. So these are the two main pathways. The first one is the 16S rRNA, where you get OTUs. So the OTUs are mapped onto a phylogenetic tree. You can say that this group of genuses, and not really at species level, but with there are databases you can correlate with to say functionally that, okay, these are the bacteria that I'm seeing, and these are the likely functions of the bacteria, and these are the metabolic pathways that are affected. And the second part is the shotgun sequencing, when you can actually look at the mRNA and look at actual expression of the bacteria as well as metabolic pathways. Then I think I'll skip the slide. And if you need to read those microbiome studies, you'll have to understand how these the data is represented, like as alpha diversity. Beta diversity is between two samples, like it's called the principal coordinate analysis, where the middle one on top, where it's comparing two samples. The right corner, top right corner, is the taxonomic distribution. So it takes a little bit of time. It's not very difficult, but to understand what these pictures mean. And the right bottom is the phylogenetic tree. So it will show you how your sample is distributed phylogenetically, whether all the samples are closely related or there is a good spread. Okay, so that is my baseline information on how we study the microbiome. Now we'll go to dysbiosis in CKD. So if you think of dysbiosis in CKD, you can think of two old men who are injured, who are still fighting each other. It's like there's a movie called Mr. and Mrs. Smith with Brad Pitt and Angelina Jolie. I don't know if you've seen it. So they're both in one family, but actually both of them are secret agents working for two different agencies. It's a bit like that. Same body, your gut is fighting the kidney, and the kidney is adversely affecting the gut. So there are certain bacteria which have been shown to be increased in CKD, and certain bacteria which have been shown to be decreased in CKD. You don't have to remember the names. It's too complex. It's when I see these lists, I think of it's like trying to remember the names of football players in the Polish football team. You cannot remember it. But metabolically, the two main groups of metabolites that are affected are, one is short chain fatty acids and two is protein bound uremic toxins. I know a lot about short chain fatty acids. I don't know so much about protein bound uremic toxins, but the three main protein bound uremic toxins that are constantly coming up in all the studies are indoxyl sulfate, indole 3-acetic acid, and p-cresol sulfate. And there are a long list of adverse events, adverse immunological effects that these have, primarily to do with pro-inflammatory state, reactive oxygen species, oxidative stress, pro-fibrotic. So all of them you can make out will lead to progression of chronic kidney disease. Short chain fatty acids are really superstars in the colon. You know, when we look at short chain fatty acids, multiple functions, lot of benefits. Short chain fatty acids are derived mostly from non-digested fibers, fruits, vegetables. They reach the colon. They are acted on by butyrate-producing bacteria. The good one is, there are many, but the one with the most benefits is butyrate. And unfortunately, patients who have kidney disease are often told not to take fruits for other reasons. So short chain fatty acids, there has been many functions. One of them is that it's involved in sodium coupled water transport. So if you have more short chain fatty acids in your diet or in oral rehydration solution, then you'll have better absorption of water. It is the nutrient for colonocytes. It is the energy source for colonocytes. It's not glucose. So colonocytes live on butyrate. And actually, glucose is not a preferred substrate for colonocytes. It's got an anti-proliferative effect, probably because if you think of malignancies, they are all very glucose dependent. If you do FDG PET is glucose. All of them require a lot of glucose to survive. So if you are not using glucose, then it's got an anti-proliferative effect. It's got a lot of effects on the immune system, mostly to develop a state of immune tolerance rather than a pro-inflammatory. It's got effect on bacterial translocation, making sure the mucin layer is adequately preserved. So multiple functions. So the short chain fatty acids, I think one of our proudest publications is where this Dr. Ramakrishna had hypothesized that adding amylase resistant starch to ORS will improve absorption of ORS from the colon, and patients will recover faster from cholera-like illnesses. But I suspect that after that, from what we know about short chain fatty acids, it probably has the reason why these patients improve may not just be that, but it could also be because of the immunological effects of short chain fatty acids. In CKD, short chain fatty acid production comes down in correlation with the degree of kidney disease. So this is human data, but actually there's very little good human data, but it's been shown that especially butyrate levels come down with degree of CKD as CKD progresses. There have been animal studies where the same super ORS was amylase resistant starch was given to rats, and it's been shown that this is occludin and claudin. These are proteins that preserve the intercellular adhesion between colonocytes. So in control group that is high, in CKD it comes down, but when CKD rats are fed with amylase resistant starch, then the tight junctions are preserved, so there's less bacterial translocation. Effects on multiple immunological intermediaries like NF-kappa B, nitric oxygen synthetase. This is monocyte chemoattractant protein, COX. All of them you see that by feeding amylase resistant starch or increasing short chain fatty acids, the inflammation comes down. And it's been also shown by looking at the histology that you can attenuate the effects of rat model of interstitial nephropathy by giving amylase resistant starch. The question is always what came first. It's a circular thing. Your kidney disease is going to worsen your bowel, your dysbiosis is going to worsen the kidney disease. So in kidney disease there is more ammonia production from the gut because there is more urea delivered to the gut, and that changes the pH. The pH becomes more basic because of the ammonia. There's dietary restriction, there's medication, there is decreased blood supply to the intestine during dialysis, even peritoneal dialysis because of increased intra-abdominal pressure, there's decreased flow of blood to the intestine. And I talked about the good bacteria and the bad bacteria. There is more protein fermentation, more production of uremic toxins in the gut, less production of short chain fatty acids. There is more inflammation in the intestine and more leakage of bacterial products and toxins into the systemic circulation. So all that produces a constant systemic inflammatory state that induces inflammation and fibrosis. I'll come to the third part. What can we do about it? So, again, one basic list of terms. We know what a probiotic is. There are many probiotics available, probiotic drinks, over-the-counter probiotics. Amul has got probiotic curd. That's probiotic. Prebiotic is nutrients that aid the growth of these good bacteria. A symbiotic is a preparation that has got both a probiotic and a prebiotic. There are two other terms which are coming more and more nowadays, is something called postbiotic, which is sometimes it's not the whole bacteria itself, but bacterial components may be enough to produce these changes. The advantage of a postbiotic is, or rather let me say, the disadvantage of a probiotic is often it has to be stored in the fridge. It's difficult to transport because they are live organisms. But a postbiotic, you can just ship it, store it at room temperature. Postbiotic is products of bacteria like short chain fatty acids like butyrate. And the last one is fecal microbiome transplantation, where a non-disease donor stool is used to change the microbiome of a patient. The problem is there is a lot of data in animal studies. The data in human studies is very conflicting, even in GI diseases like IBD. So there is only one FDA-approved indication for fecal microbiome manipulation. That's it. There's only one approved indication. Can anybody guess what it is? It's you probably come across it, but it's recurrent C. difficile. That is really the only approved indication. No other indication has been approved for fecal microbiome transplantation. So now there are two other. Fecal microbiome transplantation is difficult to do. Now it's available as a ready-made enema. It comes in a sachet. It's called Rebiota. And there is another preparation which is bacterial spores. So spores can also actually be stored at room temperature. So there is one more. So two products which have been licensed by the FDA recently. We've been using this drug called VSL3. I think you may have seen it, but it's something that's been used for pouchitis. But here, the data is also not very strong. Many studies which came out of Italy first said this was fantastic for pouchitis, but many studies subsequently have said it's not useful. The British guidelines don't really recommend it. They say that you can try it, use it for 1 month, and then if the patient wants to continue, NHS doesn't pay for it. The patient has to go and pay for it and buy it from the pharmacy. So, so in CKD also, the animal studies are quite encouraging. But if you look at human studies, the data shows that you will see improvement in CRP. If you look at the uremic toxins, it will improve. But actual conversion into mortality benefit or improvement in GFR is really not been shown yet. So if you look at meta-analysis, the bottommost forest plot is for GFR. The other ones are, I think, one for uric acid and one for creatinine. And if you look at these three studies, it's really pulled to the left side by one study, but three studies have not really shown much benefit. I know there is one tablet called Low Bun that's stored in the pharmacy here. It's a fantastic name. No, if you read it, low, that's it. They can lower your blood urea nitrogen. It's a bit expensive. I am not convinced, looking at the data myself, that it is useful. But you might have a different point of view. A Cochrane review was available from 2023, which also said that there is not enough data. The data is very heterogeneous. That's also a problem. no single study, no two studies have used the same probiotic in the same combination for the same duration. So very heterogeneous. So they've said finally there is not enough evidence to say that it works. Yeah, so that's my talk. I'll tell you one more small example. So of the problem with the probiotic. One of the problems that I think it's important to mention, because we think that probiotics are absolutely safe. That's the general impression even in literature. There was one study that was prematurely stopped because probiotics, one of the problems with acute pancreatitis is bacterial translocation. They have infection of necrotic collections in the abdomen. And one study was stopped because probiotics actually worsen the infection. Probiotics increase the mortality in the study, and one trial had to be prematurely, acute pancreatitis trial had to be prematurely stopped because of high mortality. It was a probiotic study. I think that's it. Thank you very much.
Thank you, sir, for your illuminating talk on gut microbes and chronic kidney disease, highlighting this intricate bidirectional relationship between the two. Your insights have provided us with a fresh perspective on this evolving area of research and its potential clinical implications. I have a few questions from the audience. And the first question being, with so much variability with respect to age, place of origin, etc. in this, is there a normal human microbiome? In other words, in states of no obvious gut symptoms and in state of good health, how would we recognize if a bug is causing pathology? Also, can changes in microbiome predate a pathology?
I think that question people have been trying to answer for so many decades. I don't think there's a clear answer. If you come to, we have patients coming from West Bengal for treatment, and they take a room in Gandhi Road. So by the time they get the appointment, it's about 1 week. And do you think the microbiome in West Bengal is the same as 1 week of stay in Vellore? No. The microbiome will change. In 24 hours, the microbiome will change. So there is no such thing as normal. Even when you travel abroad, your microbiome will change. When you come back to India, it will change. So maybe we could do something on people going on sabbatical when they come back, when they land one microbiome and 1 week later no. I think that it's too conflicting. The data is too, there is no normal.
Yes, sir, we do understand that it's a very vast field. So the next question being, has the example of mice microbiomes been replicated in humans? Is this going to become a future tool for weight loss rather than a bariatric surgery?
It's not, somehow it's that replication is not happening. Even there's one interesting study on autism in microbiome and the correlation with autism. So there are some features that mice can demonstrate some autistic features when they are fed microbiome from autistic children. And one of the things that it was very exciting for parents who had children who had diseases on the spectrum, but it's not come into really it's not come into standard guidelines. I think recently I was talking to the neurologists where they said one of the things they do for patients with refractory seizures is to give them a high ketone diet. I don't know if I'm not exactly sure what the mechanism is, whether it's something to do with the brain using more ketones or whether it has something to do with a change in microbiome. I'm not sure. But even in neurology department, I know that 2 people use it, but 5 people don't use it.
So the next question, is there some data to show uremic toxins may be slightly reduced with the use of microbiota changing biotics?
Yeah.
That you have covered, sir, I think.
That is there. It shows that the toxins are coming down.
And sir, is
concept about Clostridium difficile. Much before all this happened, one of our late professors of surgery used to use enema mixing normal stool for patients who had post-op very difficult control diarrhea. People used to say, what a terrible thing to do, because you used to collect stool from normal patients, mix it up and give it as enema to patients who are having refractory. It's not nothing, I mean, now you've probably got more of a scientific basis for doing it, but we used to do this in the early 70s actually, so 50 years ago. The second is about these I was involved in a study where we actually gave a combination of bacteria plus a proteolytic enzyme, and we were actually have shown very significant drops in both endocrine sulfate and cresol levels. As you say, whether it translates to improvement in renal function is yet to know, but certainly large number of patients are on this combination at the moment, and significantly able to delay the need for dialysis. See, it's not that you're going to cure the chronic kidney disease or anything, but I think it's expensive, but at least it's not as expensive of dialysis, and we're able to keep them going with conservative management a bit longer than you would without.
So the next few questions are about the use of probiotics in patients on immunosuppressants, like the patients who have received solid organ transplants, or our chronic GN patients who are getting immunosuppressions. Can probiotics be used in those patients?
Sometimes you also see patients who are on a combination of antibiotics and probiotics. So is that good? Is that really going to benefit? Is that going to kill off the bacteria, or should we give antibiotics along with probiotics? Sometimes you will see not just bacteria, but other yeasts also, fungi also, like Saccharomyces boulardii and all are part of probiotic preparations. So is that a good way to do it? Coming back to the pancreatitis study I mentioned, it is really an immune suppressed state, so in that situation, patients did badly with probiotic. With probiotics also, I guess they can also kind of overtake the normal bacterial flora. Their metabolites, their toxins might also have maybe translocating. I'm sure it's translocating. I think normally we have bacterial components in our blood all the time. So does that harm? I would not use it unless there is clear benefit. If the question is, will I use it in a patient who's immunosuppressed? I wouldn't.
What is the optimum dose of prebiotics and probiotics to alter the microbiome, and the duration of treatment, sir? Is there any approved formulation for short-chain fatty acids?
There's one disease called tropical sprue that we used to see earlier. So there, the treatment was varied from 3 to 6 months. So we are trying to, we in the sense, I don't see tropical sprue, but it used to be very common earlier. We tried to think of it as a dysbiosis, and it could not be treated by a short course of antibiotic. It required 6 months of treatment with tetracycline, which is like a poorly absorbed antibiotic, and that is probably how long it takes to reset somebody's microbiome. Otherwise, you give probiotics, and when you stop, it goes back to the original microbiome. What is the dose? The dose is not clear. The dose with this capsule called Loban, it's got 45 million units of organisms, and the other one I showed called VSL3 has got some 118 or 116 million units. So the dose of VSL3, maximum dose is 2 tablets 3 times a day. So if you're thinking of that dose with Loban, I think it'll come to about 6 tablets, maybe 4 times a day. So it's not clear how much to give. And this particular preparation that I looked at, the studies have used up to 9 or 10 capsules per day. It's not clear.
Thank you, sir. Surely it's an upcoming topic and people do have a lot of doubts about it. We hope to get
could
into
do some microbiome studies with your CKD patients or IgA nephropathy. Or
surely, sir, a food for thought.
Thank you very much, sir. Sir, now we'd like you to receive a small token of appreciation from our department.